By the time you notice a yellow leaf, your plant has been in crisis for roughly 72 hours. The emergency started inside the vascular system, invisible, long before a single cell changed color. And the one thing most gardeners do next can make it fatal. Inside the leaf, a silent countdown has already run its course. Long before the human eye spots any change, the plant's internal networks are in distress. Chlorophyll fluorescence drops, leaf temperatures rise, and electrical signals spike. This pre-visual warning window typically spans 48 to 120 hours, with 72 hours serving as a common average. During this hidden window, chlorophyll degrades, exposing the underlying yellow carotenoid pigments that were there all along. This is chlorosis, and it means the plant is actively cannibalizing itself. Through its vascular highway, the plant makes a desperate calculation. While the xylem pumps water and minerals upward, the phloem acts as a salvage route, shifting organic resources away from dying tissue. Look closely at the lower, older leaves. If they are fading to a uniform, pale yellow, you are witnessing a targeted evacuation. The plant is withdrawing mobile nitrogen, the essential building block for amino acids and proteins, from its oldest leaves to keep the vulnerable new growth alive. Look higher up, at the youngest, expanding canopy. When the delicate tissue between the veins turns pale yellow, while the veins themselves stay a stark green, you are looking at interveinal chlorosis. An experienced gardener might initially mistake this for a simple nitrogen deficiency, but the bold green-veined map quickly corrects that assumption. This pattern signals a shortage of immobile micronutrients like iron or manganese, essential elements locked permanently in place that the plant cannot relocate to new tissue. This striking vein contrast is not an invitation to blindly fertilize, but a call to investigate. But that is only the first message your plant can send. And here is where the diagnosis becomes genuinely urgent. When yellowing spreads uniformly across the entire plant, the crisis has moved beneath the surface. Saturated, waterlogged soil drives out essential oxygen, triggering root hypoxia. Without air, these suffocated roots cannot pump water or minerals, mimicking the desiccation of a severe drought. But the texture of the leaves reveals the exact cause. If the leaf margins turn dry, brown, and crispy, you are witnessing salt stress or fertilizer burn. This excess salinity reverses the water potential gradient, making it physically harder for roots to draw moisture from the soil. Conversely, if leaves remain soft and green but drop suddenly at a gentle touch, the roots are drowning in waterlogged soil. Blocked from oxygen, the plant synthesizes ethylene, forming an abscission layer that cuts the leaf loose before chlorophyll can even degrade. The underground root system is now fighting for its life, gasping for breath beneath the soil. But sometimes, a single yellow leaf is not a cry for help. Imagine standing over a flourishing monstera, shears in hand, pausing. While every other leaf radiates rich, dark green health, one solitary lower leaf has quietly turned gold. This is natural senescence, a programmed retirement. Inside its petiole, a shifting balance of auxin, ethylene, and abscisic acid gently directs the plant to reclaim its investments, shipping mobile nutrients up to the thriving canopy. A wise gardener resists the urge to medicate. They step back, leave the fertilizer on the shelf, and let the plant complete its graceful recycling. If you have ever paused before snipping a leaf like this, tell us in the comments. Did you let nature take its course, or did you step in? When yellowing starts at the very tips of the leaves and creeps inward along the margins, it traces the path of a potassium drought. Potassium is the master regulator of water movement inside the plant, keeping cells plump and opening the microscopic stomatal pores that let the leaf breathe. Without it, the plant loses its hydraulic pressure. What begins as a pale edge soon collapses into a dry, scorched border of brown necrosis, advancing steadily from the oldest leaves toward the center. What this pattern reveals next surprised even professional botanists. Look to the very top of the plant. A flush of pale, washed-out yellow new growth appears, suggesting a breakdown in chlorophyll production. Because magnesium is the central atom of every chlorophyll molecule, it is easy to assume this is a magnesium shortage. Yet, botanical science demands a closer look. Magnesium is highly mobile. When it runs low, a plant normally steals it from older, bottom leaves first, leaving them yellow between dark green veins. This pale top canopy contradicts that rule. This remains a provisional warning, a biological mystery that requires targeted testing to confirm. Beneath the leaves, hidden within the stem, lies a silent highway where a different threat can unfold. A fungal pathogen can invade the roots and creep upward, spreading silently for days before a single visible signal reaches the surface. As the fungus multiplies, it physically blocks the xylem, choking off the plant's internal water route and starving the foliage of moisture. This internal blockage produces a confusing mix of outward clues: asymmetric decline, unexplained wilt, or subtle brown streaking along the stem. While agricultural studies of crop diseases often show a 5 to 10 day lag before symptoms appear, botanical research does not establish a universal latent period for ornamental houseplants. You cannot confirm a fungal infection simply by looking at a yellowing leaf. The stem may be carrying evidence the leaf cannot yet display, but a suspicion is not a diagnosis. If a plant continues to decline even after you have corrected its watering, soil, and light, the issue is likely microscopic. Resolving this biological deadlock requires a professional pathogen assay or a plant clinic. This leaves the gardener with a high-stakes choice. Do you attempt to treat a phantom enemy, or do you look beneath the soil into the roots to make your decision? And here is where the diagnosis becomes genuinely urgent. Reaching for fertilizer to cure a yellowing plant seems helpful, but when roots are rotting, this quick fix turns lethal. Instead of saving the plant, you inadvertently trigger a secondary disaster. Damaged, oxygen-deprived roots lose their ability to absorb nutrients. Adding soluble fertilizer to saturated soil raises the electrical conductivity of the rhizosphere. This sudden spike in salinity reverses the natural flow of water. Instead of drawing moisture in, the compromised root cells face intense osmotic stress, which pulls water out of the plant and dehydrates the remaining healthy tissues. Fertilizer itself causes no harm, but applying it to a failing uptake system accelerates decline. The observant gardener must break the habit of automatic feeding. You must put down the bottle, step back from the temptation to medicate, and look beneath the surface to inspect the soil and roots directly. To save the plant, you must decode the evidence systematically. Before applying any treatment, trace a 4-part baseline. Record the leaf's location, the vein pattern, the tissue texture, and how fast the discoloration spreads. Next, check the soil moisture and pot drainage. If visual clues conflict, tools like pH strips and electrical conductivity meters can expose invisible chemical blockages. Remember, no single symptom is diagnostic across every species. Once you intervene, do not wait for a yellow leaf to turn green again. Real recovery is quiet. It shows itself in stable vigor and the emergence of healthy new growth. What color are your leaves right now? Real gardening isn't guessing, it's listening to their silent language. Subscribe to decode your plant's hidden science with us every week.